Yes, good afternoon. And today we are not specifically learning about any particular topic, but I would quickly want to go through some topics that I would, some important things in light of the assessment or the final examination, and also the internal marks. So if you are aware, we already finished several of the um, assignments, graded ones. I think so far we are right now somewhere around, you have submitted 6 assignments, if not all. I mean, differentially, you have 2 more to go, 7th and 8th. So as I can see from the grade sheet, majority of you have already gotten maybe, or if not majority, most of you have gotten 25 or so. Some people may not have finished. So I am not going to enforce you to write, submit all the assignments, and neither can I. And we all understand all our limitations. And towards the end of the semester, things get little tough for everyone. So if it is your choice, you are supposed to calculate your and ensure that you are aware of what your scores are and ensure you're getting the maximum possible you can. So once you have gotten your 25, if you're getting 25 out of 25, well and good. And it's up to you whether you want to submit the rest of the assignments or not. I would certainly encourage you to submit, because the exercise of it is about we becoming proficient in giving online lecture even. Okay, at the level of BTech, if you have started presenting a paper and presenting your views, and I'm sure you will become a good teacher in the future, a good effective communicator. Say, for example, if we were in the classroom, it is much more effective, because there is reinforcement from teacher to the student, and from the student to the teacher, or learners to the teacher. Because of that reinforcement or reciprocation, the things get much effective. Each of us can boost each other and try to improve our presentation. In an online presentation, such as recording a video, where one has to imagine the audience, are actually encouraging you, or reinforcing, or reciprocating your enthusiasm, and that's actually a tough job. As I experience it all the time. And video based lectures were given to give you that confidence and skill of being able to being self-sufficient in communicating your ideas. Okay, so if you're getting 25 out of 25, well and good, and you want to submit more, you're more than welcome. But otherwise, if you do not have enough time at this stage, so you want to restrict yourselves, I wouldn't mind either. But it's your job to get your marks. Okay, be mindful of it, and please communicate the same information to others who are not present today. So that is about the assignments. Okay, the second one I would like to talk about is the syllabus. So if we look into the syllabus, this is the syllabus that we have. It's not the one that I circulated earlier. Apparently, from your next batch onwards, the new syllabus will come into effect. Until then, we are going to follow. There's not too much change, only a slight little bit of change. So I have a few topics left here and there. I was looking through, and if I have left any more, you can please get back to me, and I'll try to finish those as well. I have race, that is rapid amplification of complementary cDNA ends. We have 5' race and 3' race. That is one topic I have to discuss. There is another topic, differential display. I will also discuss that part. If there are any others, you should be able, please try to let me know. And phage display libraries. there is the third topic in third unit. I mean, in third unit, there is one of the topics that I haven't discussed yet. And the majority of the things have been, I think most of the other things have been finished. So, and in fourth unit, the last one, we have a topic, it's a research paper. We are not going to all the details, but we will learn about how CRISPR screen can be used to identify HIV host dependency factors. That I will take in this week. So I would need a little bit, let me finish off the other stuff in the meantime. So I hope you have learned a lot from all the topics that we have discussed so far. You can also make a list of topics if there is something that needs to be redone. But before that, I would want you to watch the videos once and the respective video, and then come back to me so that I can try to discuss that again in a different way, hopefully. So before I move further, I would also want to bring your attention to something. In the beginning of this course, I have described the learning outcomes of this course. So you have to remember, I will in a short while put up a PowerPoint presentation, and you probably will be able to see what I mean by these. The first unit is about design and evaluate the best cloning strategy and molecular tools for the construction of recombinant DNA. So it is about it is about application, right? You have to all the tools are given to you, and you are supposed to engineer a recombinant DNA that can cater to the objectives that are given to you, or constraints that might be there. Similarly, you also have to have interpretation of DNA sequence or gene expression data, like we did with northern blotting, southern blotting, western blotting, something like that. Those kinds of one has to know principles, but also should be able to have skills to interpret the data. Okay. In majority of the techniques, if not all, I have strived hard to incorporate the way of interpreting. Many people can talk about principles, but once you give it data, that is when we know whether the person is an expert or not. And interpreting data is not a big deal, actually. It is just a way of looking at data. It's a way of deducting what the data says, whether the data is good, bad, or whether it's positive or negative to the given hypothesis. Those kinds of information is what you're supposed to squeeze out of what is available. And the third unit also has the same thing in terms of designing genetic construct for gene expression, when you want to express, where you want to express, and so on. And in the last one, you have same designing for a given ailment. In the last, I think assignment 8, I have given that design a CRISPR using a CRISPR, design a strategy for any disease. That is up to you. You can go to literature, find out, and then present. Even if you have already scored 25, I would suggest that you try to take it up. That will better your experience with the whole course, and also as a skill, you'll feel much more confident when you're studying, when you go for higher education as well. And so these are the outcomes. So in an examination, or even the internal examinations, I am supposed to evaluate your understanding or your knowledge of gained through this course based on what outcomes are listed here. So if you look at the assignments, some assignments are like answering CA questions. There also you have conceptual questions. But in all the cases, I hope you can see that the trial is you have interpreted several times a data. In many of the cases, you are able to design recombinant DNA technology. I mean, recombinant DNA for the purposes that is said. You also in the last two, three assignments that were given, you are supposed to discuss papers, pick a paper and discuss 2 or 3 results from that paper. So in the internal assessment, at least, we have actually reached a very high level of cognition. I will step back and take a moment again to come back to you with this. Let me just start with this 1 second. So I hope you are now able to see the PowerPoint. Okay. The how I would like if it were in a regular classroom is this, that there should be something that I'm trying to encourage you. I don't want to enforce anything, and that is the idea we always had. Even if it were in the class, that is what I would have done too. So my job is to have only two things in my teaching. That is, one is to encourage you, and the other one is to assess you. Okay, and to provide you all the materials that is required, and then hopefully and encourage you to do your best and raise above your general standards. And then I'm sure each of you must have gained a lot trying to fulfill the requirements, such as the internal assessment for this course. And the problem, the point in often is that the learning pit, they call it as, this is a concept of what is there. We should actually have, even if we discuss now, it is never late to about it. This is the start point of learning, and this is where we end. This is the target of learning. Initially, when we start learning, we find it difficult, especially when we are learning something new that as we don't have an inkling about. We don't understand. We feel a little bit, this is where we usually give up. And that's what the mistake happens often. But if we can continue, if we can hold on, and from here on, there is this is a free fall. In this case, where we don't understand, we feel, and then it is easy to just quit and not enjoy it. But if we can hold on a little longer and try a little more, I think that is where we are actually going to reach an understanding of the topic, however difficult it is. It is about our patience is one thing, to hold on to the feeling of this, the negative feeling, and a little bit of self-encouragement to strive a little more, so that we can, from there on, we are climbing up and we will reach the goal. So this is a time, it's almost like we are ending the semester. Probably it will be some 7 or 8 classes, I guess. So I would want you to revisit everything that we have discussed. If there are any difficult topics that we can always, we'll also have a bit of revision in these 7, 8 classes. So I want you to come up with topics that we need to revise. I don't want to take this particular class at the last one, right? That's why I'm trying to do it some 10 classes or so ahead of schedule. So please let me know so that we will have a discussion. With regard to the outcomes that I have shown you and the learning that we have, in typical learning is this: remembering. What is a recombinant DNA technology? What is chromosome walking? If I'm asking you that, I'm trying to make you recollect what is meant by chromosome walking. And how does it work? If I'm asking, then it is about understanding level. Majority of our questions in most of the subjects, in all my education actually, happen to be in remembering and understanding. All our schooling is mostly on remembering and understanding. And this is time now, the changes, there are a lot of changes in pedagogy that we are trying to push to applying level. This is the maximum we actually reach. But now we want at the level of creating, evaluating, analyzing. Creating is the highest level. Okay. If you look at the outcomes, majority of the outcomes are at the level of excellence. And that is one of the reasons why it is supposed to be little more critical than normal genetic engineering. All biotech people have taken genetic engineering. And I'm not sure how much you are young, and it's a little bit of basic in nature at that for your that course. rDNA is almost repetitive, except that the level of learning is supposed to be excellence. So I would suggest that you revisit all the videos, come up with questions, and then we should have a proper discussion of it, so that anytime in future you are successfully able to read a research paper that has, say, genetic analysis or construction of recombinant DNA, I would want you to recollect how much you worked during this course. And those will be the fruits of all the hard work and all the video lectures that you have done. And I will come to that. So I hope you understand that the question paper in the final exam that is likely to come is going to be, will evaluate you for your excellence in the subject. It's not going to be just mediocre. It's not simply going to be about remembering, understanding. At least it will, the base will start with applying, and then onwards you'll have other levels of doing it. Okay, you're supposed to exude that confidence and excellence in the particular topic. Okay, and with if you are listening, I mean, this is a learning pyramid. I would want you to know this, so that I hope you will feel good about going through the exercises, assignments, and classes in this course, and many other courses too, probably. So if teaching is, if you hear a lecture, you are likely to remember something about 5%, or learning could be about 5%. If you're reading by yourselves, then you're likely to remember 10%, because you have to read, you have to visualize and the things. And if you're using audio as well as visual, where sometimes some things are difficult to imagine, and there if somebody has provided you an animation, probably you are able to learn more things. With demonstration, like an experiment, you're probably like to learn more. Discussion, group discussion, 50% of learning, and practice by doing. And the most important one, and I believe this strongly, I believe it with my own experience as a teacher, I learned more as a teacher than all of my learning as a student combined. Because for every lecture we used to prepare a lot during at least when a topic is new. And that's why because we have to teach, it makes us more proficient. We have to be at a higher level than we actually can deliver. So if we are teaching others, then we have made something like 90% learning of whatever topic we have been discussing. And I believe that sorry, and that is one of the reasons or motivations for video-based lectures. There are two things about video-based lectures. First thing is that everybody should have performed, even if you are copying, not that anybody did, but If you know the concept and you're able to deliver it, you deserve the marks. Sorry. The second thing is to give you the confidence and skill of being able to communicate what you have learned, communicate a complex idea or an experiment or results. So I hope you all appreciate that, and I hope you're proud of yourselves about it. And now, before going further, please note down if I haven't mentioned it here. So the exam pattern is something like this. There are two parts, part A and part B. I think I already told, but I will mention it here so that you are clear about it. There will be, you have to answer 4 out of 6, and each is 20 marks. So total of 80 marks. So likely it will be one question could be from 1 or 2 units, mix up could be there. So if you're, there is no scope for, in this course, there is no scope for selective reading as such. So please be aware of that. And in part B, there is one question, no choice, and it carries 20 marks. So that's the total of 100 marks. So the internal assessment, internal marks could be will be calculated for 25%. So these are all tentative. Please bear in mind many things could change depending on the situations. And NSEM marks will be converted to 75%. So the total of it will be your marks. Okay. So what I'm now going to do is give you an example of 2-3 questions or 4 questions, and I would want you to, I'm not going to answer anything. You can take a snapshot of these, and I didn't include all the specific details that a question might be prepared. So I will just give you a rough idea about the questions. Okay. First question, it is a viral pathogen. It infects immune cells. It integrates into the host genome as a provirus, can remain dormant in the body for years. Occasionally gets activated, resulting in severe immunodeficiency. I think it's pretty straightforward. It is HIV, right? Now, a disease situation or description will be given like this. And then you are supposed to, the questions will be asked on sub questions will be like this. Give names and principles of techniques for diagnosis of this subject. Say if an individual is given, we want to know if that person is diagnosed with HIV or not. I mean, if he is infected with HIV or not. How do you do it? What are all the techniques that can be done? At least 2-3 techniques you should do. Say, in this case, we could have tested for the presence of antigen or presence of antibodies against antigen, or we could have done a genetic analysis like DNA, trying to find out either RNA or DNA of that particular pathogen in the blood sample, or DNA isolated from the blood. Right? So second question is, how can we cure a patient with this disease? How would you do that? Curing means you have to eliminate the pathogen from the body, and that's a difficult one. We can, with HIV, the problem is the chemotherapy, even if we use, because it can stay as a provirus and dormant for several years. It's difficult to get rid of it. That's why there is no cure in a way. Then how can we cure it? That means we have to eliminate the phage or the provirus from the body. And how would you do that? Maybe it is possible that one can do it. Maybe it is not possible. I'm not going to tell my opinions on this, but I will give you these questions to you, and you are supposed to think on those lines for a several set of diseases or conditions. The third, this is about, you can, in the next one, how can we immunize individuals against a disease like this? This is a little bit of a difficult one, especially in this case. You need to think about it. But otherwise, a general scheme can be given for how antibodies against this virus can be made, at least. You have subunit vaccines and many, many other things, recombinant vaccines and so on. And the last question here, I like this particular question all the time, is describe the limitations in your approach. Anytime, I mean, there are several individuals. All the individuals can be put into two categories. There are people who can examine themselves, and people who cannot do that. Most critical thinkers can examine their own self, their own thoughts, their own ideas, and test them. They try to find the limitations of it. And that is when they don't need an opposition to refine their idea, because they're able to fight their ideas by themselves. And that is how any idea is usually refined. So I want you to also have this capacity of trying to find the shortcomings or limitations of the approach. So so I would want you to, in all the cases that you may have given, you may have provided an answer, try to also find out the limitations of it. And that is a good practice for self-examination. I mean, critiquing your own idea. Okay, that is scenario one, or question paper one. Question number one. I have made 3 or 4 questions so that you will have a fair idea about how to prepare for this particular end semester for this particular course. Second one is, it's a genetic disease. A couple, both carriers, that means they are heterozygous, intend to have children that are free of the genetic disease. How can they have it? So one is, you're supposed to think within the scope of recombinant DNA technology, what all things we have learned. Names and principles of techniques for diagnosis of the subject. First is about diagnosis. It is a genetic disease. Assume it is a single polynucleotide morphism. Then how would you do single nucleotide polymorphism? Sorry. So what are the methods that are available, and how could you do? It all depends. Many things are there. You can test it in the level of proteins or at the level of DNA, and using southern blotting, restriction enzymes, or PCR based. Many techniques are at your disposal. And maybe you are supposed to mention 2 techniques and write up the principles of them. That is one of the things, that is what you're supposed to think. I'm not going to ask you straightforward, what is it? A situation is going to be given, and you are supposed to pick the tool that you require for the situation. And then suggest different strategies through which the individuals can have healthy progeny. In this, for example, there is one thing is one can repair the genes of this couple and then have children, or they could have in vitro fertilization, and then one can do diagnosis of the embryos and see which one are free of these mutations, and then implant them for making a baby. Or once the baby is born, you can probably, once the embryo is made, in the early stages of embryo, you can use some genetic methods to repair the cells so that the baby is free of the mutation. So there are ways of how you can do it. But then in all these, approaches, you should have used proper tools and the precautions to ensure that the born baby is healthy. Here we are talking about a human and we are talking about a baby, so there is no scope for mistakes. If there are any risks, usually we would not, it would not be allowed to start with, right? If a genetic method is made and it's going to lead to, or it is going to lead to cancer, then it's not a good idea to have that, isn't it? So that is again, when you describe your limitations in each of these cases, you probably will have to dig into those kinds of aspects. And question number 3, it's a type of cancer. A type of cancer is caused due to overexpression, and it's epigenetic. Overexpression of gene X and a mutation in gene Y. There are 2 causes. And then the questions would look like, describe the techniques to find out what kind of epigenetic changes are there in gene X and what are the consequences of it. Right? One has to, I think you know what to do. Epigenetic here, majority of the things are about DNA methylation or histone modification. There are others, but we are not discussing in our course, so we'll limit it to, mostly it is about DNA methylation. So you should know what techniques are used for finding out DNA methylation and whether it is hypermethylated or hypomethylated. That is one of the questions. I mean, that is what you should think of. The other one is about in gene Y, what kind of mutation it is. Is it a single polynucleotide, single nucleotide polymorphism, or is it large scale or chromosomal duplication, inversion, any of those? How would you find it out? Okay. Then the next question is, describe how the overexpression of the gene X could be controlled. That means we should, there are 2 things. We should reduce the transcription from gene X, that is one of the issues, or we should eliminate the transcripts of gene X. There are 2 things, right? If you want to reduce, how would you approach the problem with? If you want to destroy the transcripts of gene X, what could you do? I hope you know with CRISPR we can do epigenetic changes, right? Like we have discussed in the last class, we could have made a translational fusion of a DNA methylase because we want the methylation of the promoter region. We could have had with a DNA methylase translationally fused to a defective Cas9 and use CRISPR RNA that is specific to or single guide RNAs that is specific to the transcriptional or regulatory region of gene X. Then what would happen? DNA is likely to be methylated at that pace, and because of which transcription could be reduced. The other way is to use RNA interference. And if we didn't, I'm not sure if we discussed it. If not, put it on the list, and we will try to discuss RNA interference as well in the next some other class. The next one is describe how the mutation can be corrected. If you remember gene therapy or making transgenic animals, those 2 come into this place. Or you can also use CRISPR-based methods, right? You have to do, one has to cut the place where the mutation is and then provide a donor DNA with a correct sequence, which would encourage homology-directed repair, and then the genetic defect is usually corrected. And again, there are a lot of limitations for each of these approaches, and you have to think about the limitations. And question number 4, I think you are all much more aware of this. A fungal organism produces a novel amylase. However, the genome sequence is unknown. We don't know any genetic genome sequence of it. Describe how to isolate the gene that encodes the amylase enzyme. We here it is about construction of genomic libraries or cDNA libraries that you have to figure out what kind of host should be used and what kind of screening method should be used. You know, amylase, starch. I think you have done a lot of experiments in your industrial biotechnology or somewhere. Um, you can add starch, iodine based. You can see decolorized zones, and that is how you can figure it out. So, but here the focus, of course, you have to have the screening method properly. You should have known which enzymes are what kind of host is best for it, and so on. And the second one is, say you have isolated the gene that is encoding amylase. Then how would you produce it in large quantities? I want to produce a large quantities of amylase enzyme. So how would you do that? Which organism would you choose? How would you overexpress it? How would you collect the protein, and so on? And again, the same considerations that are to be borne in mind. If it is a prokaryote, what is a host? Is it prokaryote or eukaryote? And here I gave you a fungal gene. It is an eukaryote. Can it be overexpressed and produced with the same processing as is possible in? I mean, if it is possible to do that in a prokaryotic host strain or not? So one has to think all this stuff before you even propose your ideas. Okay. So I think I have given you a fair idea of all the questions that are likely to come. Majority of the questions will be at this level. Okay. The terminology or the wording may not necessarily be the same, but in essence, the question will be asked somewhere on these lines. You might also have to remember that there'll be much more specific details of the genes and other things. If I give you more specific details about the nature of the gene, organism, disease, or enzyme, and so on, then your answer should become highly specific according to the given constraints. So right now, what I tried is to give you a fair idea about the nature of the questions. If you have already solved the CAA questions in the part B of CAA question paper assignments, then you know what kind of the specificity with which the questions are likely to be asked. So keep that in mind as well. So far, I want to say this. I'm very proud of you people. Majority, if not majority, at least half the class have showed clear intent of learning and performing. And many times I saw your videos, and they're amazing actually. If you keep continuing, if you continue these exercises a little more, I think by in a year or so, you are ready for PhD, not necessarily just PG. You can take up a PhD right after your BTech too. So your capacities have increased. All you need now is to probably relearn all the concepts that you have learned from cell biology, molecular biology. One has to start with biochemistry, cell biology, molecular biology. These are the basis for everything else. Of course, genetics and everything else also will add up. So once you have finished this semester, I would certainly encourage you all to go back to your fundamental books, biochemistry, cell biology, molecular biology, genetics, and read them once again. And I'm sure you can qualify any of the examinations. And once you qualify, you can directly take up a PhD. So try to think on those lines. You are supposed to pat on your back, be proud of yourselves. And I'm certainly proud of your videos and the efforts that you have put. Some people have done really excellent. And I didn't probably mention it. Sometimes I've mentioned, but not all the time. But we are all here a unit. So I am saying this now to you all that you have done a great job and I'm very proud of you. Okay. So I will stop here, and if you have any questions, you can ask regarding any of the examinations or whatever we have discussed today. And you can, if required, you can also give me the list of topics that you want me to repeat or take up in the next classes before the examinations. Any questions, please? Regarding internals, assignments, any questions?
So it's not about the internals, but about the final exams.
Yes.
We haven't gotten
Yes.
any other information.
Yeah. What exam? What's your question?
Like, sir, will we be called back to college, and around what time will the exams be?
Yeah. I mean, that is that we cannot say. It's not decided, or it's everything is tentative. But you are in final year, so if you are in final year, you will have examinations. When, where, how, we don't know. That has to be, the situations are so volatile or they're changing too fast. So we have
Yes,
to
sir.
think. I think they'll make a decision and communicate. Yeah. Yes.
Okay, sir.
Any other questions regarding? Yeah. Yeah.
So will this format be followed for all the subjects or just for rDNA?
No, only rDNA. I mean, each
Okay, sir.
faculty has to decide what pattern is coming. So you better inquire with other faculty members what is the pattern.
Okay,
For
sir.
rDNA, this is the pattern.
Okay, sir.
Okay. Yes. Any others?
And sir, this is not about exams at all, but will there be SAP next semester?
What is it? A SAP.
Semester abroad. Yes, sir.
Uh, I don't think so. Um, I don't think there will be, but one cannot say that. We don't know actually. But considering the situations, one has to make a serious backup about doing it in India as well. Because if you just rely on it and not able to find anywhere, then you don't know how things are. Because the labs outside are also not so encouraging, because it's not like taking a PhD student, right? It's a short term and the risks are high, and the outputs are actually little for all the people, in the sense that 6 months not much can be done, isn't it?
Yes, sir.
It's mostly training you. So I don't think the labs abroad would want to take any chances on that. I'm
Okay,
just
sir.
guessing. Okay, I'm trying to think of in the perspective of psychology, you can say. Yeah.
Yes, sir. I understand.
Yes. Yes. Any others? But you can also, I think, for final year project, you can try Indian labs abroad. I mean, Indian labs outside Shastra. There are some excellent ones. That might save you more time.
Okay.
Yeah, if you can start early. Yes. Any other questions? Can you tell me any topics that we are supposed to repeat, or that I might have missed mentioning?